Hi, it's Mr. Andersen. In this podcast, I'm going to talk about mitosis. Mitosis, in general, is used to mean exact copying of cells. Technically, it means division of the nucleus, but in biology, we tend to refer to mitosis as making exact copies and then meiosis as making sex cells. Why is mitosis important? Well, when you were born, you were a tiny little zygote, and you moved from that to an embryo and a fetus and to you through a process of mitosis. And when this lizard loses its tail, it's going to regenerate a new tail using mitosis. And when this anemone divides in half through asexual reproduction, you guessed it, that's mitosis. Or when you even form a scab, that's mitosis. So mitosis is important. And it's also important that we ensure that the DNA in that first cell is in all the cells that are copied from that, because that's how life works. You start life as an individual cell with DNA in it, and you eventually end up with this complex organism that has billions and billions of cells, and each of them have the exact same DNA inside it. Before we can talk too much about mitosis, we need to get this out of the way, and it's the idea of diploid versus haploid. Basically, if we're looking at an organism or a cell that's haploid, it essentially means it has one copy of all of its DNA. And so in eukaryotic cells, our DNA is organized into what are called chromosomes, or linear stretches of DNA. And so if I were to name these, I would name this as 1, 2, 3. We generally do that from the longest to the smallest. This would be chromosome 1, chromosome 2, chromosome 3. But this cell would just have one copy of each of those. And we refer to that as an haploid cell. Or it's N, or it has N copies of chromosomes. In this case, N would equal 3. A diploid cell is going to have two copies of it. In other words, it's going to have 2 of chromosome 1, 2 of chromosome 2, and 2 of chromosome 3. And so we call that diploid. And I remember di just means 2, so it has 2 chromosomes. In this case, 2n equals 6, since we have 6 total chromosomes. Let's apply that into humans. And so in humans, a sperm like this or an egg like this are haploid. And so for each of those, n equals 23. Same thing here. But a baby comes from a cell that copies itself over and over and over again. And since we're fertilizing a sperm with an egg, this is what the chromosomes in a human look like. In other words, 2n equals 46. You have 46 total chromosomes. You have 2 chromosome 1, 2 chromosome 2, 2 chromosome 3, and it just keeps going like that. Now, this is just staining on the chromosomes. Each of the chromosomes will have individual genes that are on it, and it'll be thousands of genes that are found on each of that. But you have, inside every cell in your body, 2 chromosome number 1s, and you got those from your dad. And you have you get 1 from your dad, and you get 1 from your mom. And likewise with all the chromosomes in your body, you have 2 copies of each. And so hopefully that helps. That's what diploid and haploid means. Now, the cell cycle is basically how we go from one cell, and if I were to draw that in, it'd be like one cell here with a nucleus, and how we eventually make 2 cells. And that, let's start from the outside of the cell cycle. And the cell cycle is really kind of just like a photocopy machine. Once it starts, we make copy after copy after copy after copy. And so basically what we do is a cell starts here, and the first thing it's going to enter into is G1 phase. Now that whole G1 phase right here, and it just stands for gap, but I remember G1 stands for grow because that's what the cell's going to do. So this G1 phase is a cell just making, getting larger and larger and larger. So it's getting bigger and bigger. Now this is actually the first step in what's called interphase. And so this would be all interphase. So from here all the way back around again is interphase, and then this would be mitosis right here. So basically what's next? Well, the next phase is going to be the S phase. So in this S phase, which is again part of interphase, that's going to be where we actually copy the DNA. So this is going to be where we're doing DNA replication and we're copying all the DNA. And then we're going to have G2 or gap 2, or we call this grow 2. And so these are all the phases of interphase. So basically, what does a cell do in G1? It's going to grow. What's it do during S? It's going to stop growing. It's going to copy all of its DNA. And then in G2, it's going to continue to grow. And so at the end of this, so after we've gone through all of interphase, a cell is ready to divide. And so at this point, it's actually going to go through mitosis, what we would say as prophase, metaphase, anaphase, telophase, and then cytokinesis. And we'll talk about all of the specifics of how that works. And so if you look at it, where's most of the time spent? It's actually spent in interphase. Now, there are some cells that never really copy themselves. So the nerves, for example, in your central nervous system, so the nerves in your brain, aren't going to make copies of themselves, and muscles are the same way. So they are stuck in what's called a G0 phase. But all of the other cells in your body are making copies of themselves because they wear out and they have to make extra copies. So before we can even look at the parts of mitosis, we should understand what a chromosome really is. And so this is the characteristic shape of a chromosome. It's that X shape. If we were to go to the specific parts of it, basically one side of it is going to be a chromatid. And so if you were to look just on one side of it, so this one right here is a chromatid. So this would be one chromatid. And this would be the sister chromatid over on this side. So where do we get chromosomes looking like this? This is going to be after the S phase. So after the S phase, remember, we've copied all of our DNA. And so basically, if you've got DNA over here, you have the exact same DNA over on the other side. So basically, what are some other parts? You're going to have the centromere, which is going to be in the middle. And then there's not going to be any genes right there. And then we're going to have a short arm and then a long arm on this side. But again, when you look at a chromosome like that, that's a chromosome way down here. And sometimes we get confused on, well, where's the DNA? Well, the DNA is it's that double helix. It's wrapped around these histone proteins. And then you got these beads and beads upon beads and lines upon lines. And so really, that DNA is folded and folded and folded upon itself before it eventually gets to the level of a chromosome. And so basically, it's usually going to look something like this when it's in a cell. It's what we call chromatin, so it's all loose in there and it's doing its job. It's actually making proteins. It's controlling the cell. But before we can copy it, it has to coalesce even more so it gets this characteristic chromosome shape. Okay, so now let's go through the phases of mitosis. I remember this as IPMAT. It's interphase, prophase, metaphase, anaphase, telophase. The one thing that you should remember is that mitosis is not specific phases where it's moving jerky. It's a movie that just kind of moves from interphase to prophase to metaphase to anaphase. And so there's some great movies out there. You can see cells going through mitosis and you could watch this process occur. But what I'm going to do is step you through the important phases and then what's happening there. And so let's look at this. This is a cell that's now in prophase. And so what's happened so far? Well, you can look here. We've already gone through gap 1. We've copied all the DNA, and then we've gone through gap 2. And so now we're in prophase. So what's going to happen in prophase? Well, all that chromatin, in other words, that loose DNA and proteins, you can see has coalesced together, and it's in chromosomes. There's no more nucleolus. Nucleolus, remember, makes ribosomes, and that's kind of dissolved. We have separation of, this should say centrosomes. So there's centrosomes that are moving to the side. We're going to find this in animal cells. And then we start to get the formation of the spindle. So the spindle are these blue tubes, and those are going to eventually attach to the chromosome. So let's go at the next phase and see what's happened. Now we're in what's called prometaphase, so we're moving to metaphase. So what are some things that have happened? Well, you can see that nuclear envelope has started to break apart, and then the chromosomes are forming what are called kinetochores, and those kinetochores are right here on the side. You can see one there, or you can see one there. I like to think of kinetochores almost like Pac-Man, so they basically will sit there like this. They're going to sit on the inside of the chromosome, so like that. And then the tubules that make up the spindle, let me find a different color. So if we find blue, basically the spindle are going to attach to the kinetochore, and it looks kind of like that. And so basically I like to think of this as like Pac-Man eating its way out to the other side. It doesn't really occur that way, but that's how I remember it. Let's look at some of the other important things. We've got microtubules starting to attach. You can see we're starting to get some attachment to the chromosomes. So this is prometaphase, excuse me. So we're still not to metaphase. If we go to metaphase, and I have some mnemonic devices to remember this. So during metaphase, I always remember that they're going to meet. So the M in metaphase, stands for meet. So they're coming together to meet, and they're going to meet in the middle. And so basically all the chromosomes are going to line up right here along what's called the metaphase plate. And so there's going to be this imaginary line that goes right down through the middle of the cell. And you can see that they're all lined up right along there. It's kind of like a tug of war. It's pulling on it on either side, and they're eventually lining up right in the middle. Now we have the anaphase, and so what happens in anaphase? Well, you can see that they've pulled apart, and so they're moving in either direction. So I remember that the A in anaphase stands for moving apart. So they're separating to either pole, because remember what we have to do is make sure that each of these cells have the exact same genetic material. If we go to the next phase, then that's telophase. Telophase, I always think the T stands for the end. And so we're getting close to the end. So what we're getting is now a new nuclear envelope starting to form around each of those. They're starting to unfold from that characteristic chromosome shape, and they're starting to form chromatin. We start to get the nucleoli show up, and then the cells continue to get bigger. And you can see here that we've kind of undergone mitosis. In other words, the nucleus has divided. And so what's the last step? Well, the last step is cytokinesis. In other words, that cytoplasm has to divide in half. And so cytokinesis is the divvying up of the rest of the cell or the rest of the cytoplasm. And there's important stuff in there that you can see here. We've got some endoplasmic reticulum, some mitochondria, lysosomes. That's all going to be divvied up on either side. Now, if you think about it, what's the next step? Well, the next step is this cell is going to go into G1 phase. So it's going to start growing, it's going to copy its DNA, and then it's going to grow again, and then it's going to divide in half again. Now what we're looking at here is animal cells, but plant cells will do something similar. The only difference is that instead of zipping in half, because they can't really do that because they have a cell membrane, what they'll do is they'll build something called a cell plate that's going to form right here in between the cells, and that'll eventually form a new cell wall, and eventually we'll have two cells as they split in half. But aside from that, it's pretty much the same way. In fact, here's some cells undergoing mitosis. This is just a diagram, but it's basically onion root cells. So onions, when the roots grow, they do that by making more copies of the cell mitotically, and that makes the cell get bigger and bigger and bigger. We could go through and look at these cells and try to figure out what phase they're in. So I would call that interphase and interphase and interphase and interphase, and this one I would call prophase, and then interphase. And you get the sense that almost all these cells are in interphase, which should make sense because when you look at the cell cycle, they were spending most of their time in interphase. But right here is one that's actually pulled apart, and so I would call this telophase. Or this one, let's see, they're meeting right in the middle. That'd be metaphase. Or here they are pulling apart, so this would be like an anaphase. And so basically we could count the cells and what phases they're in. We could figure out kind of a pie chart of how much time they spend in each of those phases. And this works great over and over and over again, but sometimes it works too well. And so all cancer is cells reproducing out of control. In other words, we have all these mechanisms inside our cells that stop cells from doing this. But occasionally, there'll be a mutation, and that'll suppress some of these cancer-suppressing genes, and eventually you have cancer, which is a bunch of cells that make copies of themselves. The troubling thing about that is these cells can move different places in your body, and they can activate other cells to become cancerous. And so what do we call a group of cells that are actively growing out of control? We call that a tumor. And you can see some tumors forming in this person who has lung cancer and this person who has a tumor in their brain. So you can see here, this is a graph of cigarette consumption in men from 1900 to 1940. You can see that it increased. And then this is lung cancer rates. And you can see that there is roughly like a 20 to 40 year lag between how many cigarettes they were smoking and the amount of lung cancer. And so there's genetic predisposition, but we can also have carcinogens. In this case, it's chemicals found within the smoke that can cause mutations and cause cells to jump in and become cancerous. But what is it? It's just uncontrolled cell growth, uncontrolled mitosis. And I hope that's helpful.